Hello friends, welcome to CEC Live Lectures. Dear friends, with our ongoing series on cell biology, today we are going to talk from where we left in our previous session. So we would be continuing further and would be talking on endocytosis, exocytosis and lysosomes. And for the discussion on the topic, we have with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. Dr. Dogra has immense experience as well as she is contributing a lot in the area of academics. Friends, if you wish to ask questions from Dr. Charu Dogra Rawat on today's topic, do call us through our toll-free number. Our number is 1800110430. I repeat, our number is 1800110430. All dear friends are requested to call in the last 10 minutes of the lecture so that first you could have in-depth knowledge on today's topic, and yes, if you have any queries, we will try to give answers to your questions through Dr. Charu Dogra Rawat. So now I would like to welcome our guest, Dr. Rawat, once again and would request her to give us in-depth knowledge.
Thank you. So we have been talking about the protein transport. We have talked about two organelles in detail, which is the endoplasmic reticulum as well as the Golgi complex involved in this protein transport process. Today we'll move further and we'll talk about two different mechanisms, which is exocytosis and endocytosis, the cell secretion as well as the cell uptake. And we will also talk about an important organelle, which is the lysosomes, which causes the degradation of various unwanted things which are there in the cell. So we'll look in detail, and this is basically the intracellular protein transport mechanism which we have seen. So there are different kinds of transport, just to refresh our minds with this one picture only, which is that there are three different types of transport mechanisms: the gated one, the transmembrane one, and the vesicular one. We have emphasized and studied about in our last two sessions, which is about the endomembrane system comprising of the Golgi apparatus and the endoplasmic reticulum. So we have talked about how the proteins are formed inside the endoplasmic reticulum and how they are transported to the Golgi apparatus. Now we'll move on from the Golgi apparatus in this vesicular transport region, and we'll talk about that how this late endosome, early endosome and secretory vesicles play a part, and how these proteins go out from the cell surface. And because it is talking about the protein and other transport system, we will also talk about how the cell uptakes the material from outside, and then finally we'll talk about how they go and end up into the lysosome which is there. So this is the vesicular protein transport system again, which we have talked about, and there are two processes that are involved. The process that a cell uses to take in molecules from the extracellular space by folding its membrane around it and forming a vesicle is termed as the endocytosis. So we'll talk in detail about this mechanism, where the things which are outside the cell, how the cell uptakes it. So the cell uptakes it through these vesicles only, and therefore it is called as the vesicular transport. So how the things come in, how then they fuse into late endosome, what are these structures, and then how they fuse and further with the trans-Golgi apparatus to come inside it. We'll also talk about another process, which is that a cell uses the vesicles to release the substances to the extracellular space, a process which is termed as exocytosis. So in exocytosis, we have been following this path, and we have done in detail about this path, that how the things which are formed on the surface of this rough endoplasmic reticulum when the ribosome sits in, the proteins that come inside it, how these different kinds of vesicles, which are the COP2 vesicles, they transfer it into the Golgi, transport them into the Golgi network. Then from the Golgi, it goes from the cis to the trans-Golgi network, which is towards the periphery of the cell, and then how we'll talk about further that how these molecules from here are secreted outside the cell from the plasma membrane. So this mechanism we have already talked about, the endoplasmic reticulum and Golgi in detail. The further mechanism that how these from the proteins from the trans-Golgi are then secreted outside the plasma membrane, we'll talk in detail today. Now, endocytosis and exocytosis, these processes play a major role inside the cell. The various cellular processes are such as for the hormone secretion, peptide secretion, amino acid secretion, all the secretory things which the cell needs to release it into the environment or surrounding it basically passes through this exocytosis pathway. So this is a hormone secretion, I mean primarily. There are other mechanisms, but this is the primary way by which the secretions are going from the cell to the outside environment. There is membrane receptor internalization, then there are the receptors which are present on the membrane, or there are receptors which are present on the internal organelles, and these receptors when the vesicles they go outside, then they need to take up the protein which are formed outside the cell, or the things which are released outside, or the vesicles which just go outside, the proteins, the receptors which are going on, they are also internalized by this endocytotic procedure. There is pathogen engulfment, the cell eating, cell drinking, all happens by the mechanism of endocytosis. And then there is a neuronal communication which basically involves both the exocytosis and endocytosis at synapses. So when the neurotransmitters are released from one neuron cell and they are taken up by another neuron cell, both the mechanisms are involved. So they play a major role and they are very crucial for the cell to sustain. We'll just talk about a little bit of history of this discovery of endocytosis and exocytosis to begin with. In 1882, Ilya Mechnikov, using a light microscope, observed that specific cells engulfed invading pathogens. And he called this process phagocytosis, where cell engulfs the pathogen via the vesicle formation. So it dates back to 1882, when the first time this cell eating or the phagocytosis was observed under a later light microscope. Then around 50 years later, in 1931, Warren Lewis observed a similar process of vesicle formation when cell took in fluid, a process which is called as cell drinking, or he named this behavior the pinocytosis. So phagocytosis, pinocytosis. In 1953, George Palade, while examining the structural and functional organization of the cell, discovered cave-like invaginations of the membranes, and he called them caveoli. He inferred that these must be required for the cell intake. So another, landmark point that, the cell is actually taking there is invagination of the cell that is occurring. And these invaginations or these caveoles basically are responsible for the cell intaking the material from outside. In 1955, Nobel laureate Christian Dudwig coined the term endocytosis for the first time, which encompassed both these mechanisms of phagocytosis and pinocytosis. In 1975, Michael Brown and Joseph Goldstein, with electron microscopy expert Richard Anderson, observed that when low-density lipoprotein or LDL binds to its cell surface receptor, it leads to the formation of what are called as the coated pits. These pits are then internalized and endocytose LDL receptors. This was the discovery of the third type, called the receptor-mediated endocytosis. So till 1975, they knew phagocytosis, they knew pinocytosis, and both these were coming under the endocytosis. Then further, they got to know that there is another mechanism of endocytosis, which is receptor-mediated endocytosis, where the receptor binds the ligands which needs to be internalized. In the same year, Barbara Pierce isolated the major coat protein, a triskelion molecule, if you remember, and named it as the clethorin. Therefore, this process is also called as the clethorin-mediated endocytosis. This particular mechanism, the clethorin-coated pits, etc., we have already talked about when we talked about the different vesicles which are arising from the endoplasmic reticulum as well as the Golgi complex. So we have already talked about the structure of the clethorin-coated pits, how they are formed. We have already talked about the COP1 and COP2 molecules, which are responsible for the transportation between the endoplasmic reticulum in to and fro in the forward and reverse direction. So we have talked about the different kind of vesicles which are there in our previous sessions. The discovery of endocytosis and basically exocytosis came up in 1980 when Randy Schemann's group generated yeast mutants that were secretion deficient and revealed the presence of critical genes that coded for proteins necessary for exocytosis. So he basically, identified certain mutants which could not carry out the cell secretion. the cell secretion or the exocytosis. And then he identified the genes which were responsible for the cell secretion to occur. So these mutants has the mutations in those particular genes, or therefore their proteins are not well formed, and that is why the exocytosis could not be, or basically the cell could not secrete the thing. So they were, kind of deciphering the mechanism of exocytosis, etc. In the 1993, James Rothman identified some of these proteins, and based on their chemical nature, they were called snares. In our last session, we already talked about the snares, that how this traffic, how this the lodging of the vesicles onto the next membrane takes place, or how the cell vesicle fusion takes place, and these are very important proteins in responsible for that. In his seminal snare hypothesis, he proposed that these helical structures hook onto each other, causing membranes to come closer with sufficient force to fuse and generate the exocytosis. So we have talked about the molecular mechanism and the involvement of snare hypothesis in our previous session, but James Rothman was the person who basically described all this. And around the same time, Thomas Sudhoff established that this process was also tightly controlled in neurons by calcium-sensing proteins called as the synaptoteguments that fostered and accurately timed vesicle fusion for neurotransmitter release. So as I told you that at neuronal junctions, this exocytosis as well as endocytosis is taking place. So Thomas Sudhoff basically described and identified certain proteins which were responsible, which were calcium sensitive and were responsible for accurate timing of the vesicle fusion that is taking place at the synapses, basically for the transmission of the neuronal impulse. So that is these three were the main scientists who described this mechanism, and for their contribution they were given the Nobel Prize in Physiology or Medicine in the year 2013. The Nobel Prize in Physiology was given jointly to James E. Rothman, Randy Schiemann and Thomas Sudhoff for their discoveries of machinery regulating this vesicle traffic, a major transport system in our cells. So you can understand the importance of this mechanism, the vesicular transport mechanism that was basically recognized all over the world, and it is so important for the cell, which is for the transport of proteins as well as internalization of various other molecules. So looking at the different modes of exocytosis and endocytosis and how they are coupled, we'll just quickly go through this picture, and we will see that there are three different modes of exocytosis as well as endocytosis which have been described, and at certain points these two basically they couple together to carry out the entire mechanism. So these are the three mechanisms. The one is called as the full collapse fusion mechanism, another is the kiss and run mechanism, and then there is a compound exocytosis. So these are the three mechanisms for exocytosis. For endocytosis, there is a classical endocytosis, there is a Belk endocytosis, and then there is a kiss and run endocytosis also. So exocytosis is basically when the things need to go outside. So you can see that these things are basically the regular or the smaller quantal size. This is basically telling the reference about the size of the cargo which is basically secreted outside, and it basically has been described in detail at the synapses. So a little reference to that procedures is also given. So what happens in the full collapse fusion exocytosis? It releases the vesicular contents completely and rapidly via a rapidly expanding pore. So in this fusion, there is a rapidly expanding pore which is there, and the entire content is thrown outside. For the kiss and run bypasses vesicle collapse and is often considered to have a narrow pore and may thus release contents slowly and or partially. Partially it's correct, but slowly it's, it sometimes does slowly, but sometimes again it's rapid. But it is called as kiss and run because it bypasses the vesicle collapse. So it just fuses with the, for an instant it fuses with the plasma membrane. partially the things are released inside, and then again it becomes a vesicle. So it basically does not lead to the vesicle collapse, which is found in the full collapse fusion. In compound exocytosis, very large and interconnected vesicles are formed by a vesicle-vesicle fusion, which then fuse with the targeted plasma membrane and discharge the cargo from the cell. So multiple vesicles, they tend to fuse together, and therefore it is called as the compounded mechanism. They fuse together, and then all of them, and one of them basically goes and fuses with the plasma membrane, targeted plasma membrane, releasing the entire cargo of all these vesicles into the outside or into the other cells which are there, into the outside environment which is basically there. For endocytosis, full collapse fusion is followed by classical endocytosis involving membrane invagination and vesicle reformation. So if you recollect it from this picture, then this is basically the full collapse. So if this is a full collapse fusion exocytosis, then this is the endocytosis, the classical endocytic pathway. You can see these are the clathrin pits which are there. So this is a clathrin coated endocytosis. So you can see that the membrane, there is an invagination that occurs, and then these are endocytosed basically. There is another mechanism which is called as the bulk endocytosis. It is usually followed by the compound exocytosis that retrieve the giant vesicles. So when the multiple vesicles fuse together to give rise to giant vesicles, and when they exocytose their content, then it is compound exocytosis. But because the lot of material has gone outside, it is usually followed by the bulk endocytosis when this giant vesicles are actually retrieved inside from the surface of the cell. Kiss and run mechanism is similar in both the cases. It's only on the either side of the plasma membrane. So if it is towards inside, it is endocytosis. If it's towards outside, it is exocytosis. So these are the different modes in which the endocytosis and exocytosis work. And we have also seen that, usually the fusion mechanism is followed by classical endocytosis. The compound exocytosis is followed by bulk endocytosis. So there is a coupling between the two mechanisms which is there. This coupling is very important because when the vesicle fusion is taking place, then the plasma membrane is reorganizing. All this material needs to come back inside the cell. So the plasma membrane integrity, it's a fluid thing, it needs to be maintained. So all that thing again needs to be internalized. So if the material is exocytosis, the content will be exocytosed, but the plasma membrane will be recycled. So endocytosis has to occur, apart from obviously taking up the material or uptaking the material which is present outside the cell environment. So exocytosis, this is the secretory pathway. Let's talk about the detail of the pathway now. So exocytosis secretory pathway, there are two different mechanisms for exocytosis. One is termed as the constitutive secretion, and another is termed as the regulated secretion. Constitutive secretion, all the time secretion, it's a non-calcium dependent secretion, also termed as. Regulated secretion is dependent upon the concentration of these calcium ions. So you can see that this is the Golgi apparatus and this is the trans-Golgi apparatus. These are the newly synthesized plasma membrane proteins when they are fused together, and this is the unregulated membrane fusion that is taking. So this is the default, this is going to happen, this is constitutive, you do not need any trigger for this. All the plasma membrane proteins which need to be integrated into the plasma membrane and displayed outside, or they are transmembrane, these will form vesicles and they will constitutively go and bind here. But the Golgi complex might also contain some of the proteins which need a stimulus to be secreted. So that follows another pathway which is called as a regulated secretion. The regulated secretion, these proteins will then form a secretory vesicle where they store these secretory proteins and other substances. They wait for the stimulus. Like in this case, there is a signal which is coming from the outside of the cell that something has to be released. And this signal is coming because some molecule is binding to the receptor, and then this is the signaling pathway. This is the causing the stimulus to be here, and therefore this vesicle will go and fuse with the plasma membrane and release its content outside. So exocytosis is basically consisting of two different pathways. One is a constitutive secretion, does not need any stimulus. Whatever the proteins are formed, they have to become a part of the plasma membrane or need to go outside. Therefore, it will follow a path which is not dependent upon any kind of signal which is required. And a regulated secretory pathway, which is for the proteins which needs certain kind of a stimulus. When a hormone, for example, or on any receptor, any ligand is binding, it's creating a kind of a signal pathway. We always talk about that the hormonal secretion, is regulated secretion. Some of the hormones are regulated. It depends upon the kind of stimuli we are giving to the cell. So that stimulus is basically signaled to the vesicles, and the vesicles containing those hormones or peptides or enzymes are then fused with the plasma membrane to release the things which are needed outside the cell. So the two kind of secretory pathways are there. The constitutive pathway involves proteins that are continually secreted by the cells in which they are made, being carried promptly to the plasma membrane after being packaged into the membrane-bound transport vesicles. Constitutive exocytosis is performed by all cells and serves the release of components of the extracellular matrix or just delivery of newly synthesized membrane proteins that are incorporated in the plasma membrane after the fusion of the transport vesicle. So all those constitutive things which need to go outside for the ECM or for plasma membrane follows this constitutive pathway of secretion. The regulated pathway of secretion involves proteins and or small molecules that are stored in the vesicles and only fuse with the plasma membrane after an extracellular signal triggers the cell. Unlike the constitutive pathway, the regulated pathway is found primarily in cells that are specialized for secreting their products. As I said, for example, hormone secreting cells, the neurotransmitters, the digestive enzymes, because they all need stimuli. They need stimuli, the compounds the proteins are made and stored inside the vesicles, but as the appropriate stimuli is there, the appropriate signal comes, these vesicles go and fuse with the plasma membrane and release their contents outside. And the activation of receptor generates various intracellular signals, including a transient increase in amount of the free calcium in the cytosol. That is why the regulated pathway is called as the calcium-dependent secretory pathway. These signals initiate exocytosis, leading to the fusion of secretory vesicles with the plasma membrane and release of the products to the extracellular space. So the two different mechanisms of exocytosis are there. Now the steps which are involved in this exocytosis, we have talked about the molecular mechanism of these steps when we were doing the different types of vesicles. So I will not detail about the molecular mechanism here, but we will just talk about the overview of the basic steps that are involved in the exocytosis. So the first is the vesicle trafficking. We have already talked about protein trafficking, in endoplasmic reticulum when they are formed, then in Golgi complex where the sorting takes place and they are, targeted to different organelles. So it involves the translocation of a vesicle. Once the vesicle reach their targets, they come into contact with tethering factors that can restrain them. So the first step in exocytosis is basically the vesicle trafficking which is there. Vesicle tethering, initially loose tethering of vesicles with their targets, followed by more stable packing interactions occur. Tethering involves links over distances of more than about half the diameter of a vesicle from a given membrane surface. So tethering is basically, it's a kind of a loose attachment and then can be followed by a very stable packing kind of an attachment which is there. So the vesicle needs to go and fuse with the target vesicle or to the plasma membrane, or so it needs to be trafficked along that path. As soon as it finds the target, it needs to tether. So it needs to generate these kind of attachments which will basically tether it to the target vesicle which is there. Next comes the vesicle docking. The term docking refers to the holding of the two membranes with a bilayer's distance of another less than which is 5 to 10 nanometers. Stable docking probably represents several distinct molecular states. The tethering and docking of a transport vesicle at a target membrane precedes the formation of a tight core snare complex. Again, I tell you that molecular mechanism of this formation of snare complex we talked about in the last lecture, but overall when the exocytosis needs to take place, then the tethering and docking involves or precedes this core snare complex formation that was occurring. The next step is the vesicle priming. In neuronal exocytosis primarily, the term priming has been used to include all of the molecular rearrangements and ATP-dependent protein and lipid modifications that take place after initial docking of a synaptic vesicle but before exocytosis, such that the influx of calcium ions is all that is needed to trigger nearly instantaneous neurotransmitter release. In other cell types whose secretion is constitutive, that is continuous, calcium ion independent and non-triggered, there is no priming. So priming is basically that, when all those kind of arrangements have taken place so that the content can be released instantaneously. So there are certain changes which has taken place, such as the influx of calcium ions. So when the influx of calcium ions takes place in the neuronal vesicles, immediate release of neurotransmitters occur. So there's a snap difference in between that. So therefore vesicle priming is an important procedure there. Vesicle fusion is the last step in exocytosis. The vesicle fusion is driven by snare proteins and is the process of merging the vesicle membrane with the target one, that can be the plasma membrane if it is secreted outside the cell, resulting in release of large biomolecules in the extracellular space, or in case of neurons in the synaptic cleft. So the vesicle fusion is there. So we have seen that there is vesicle priming, there is the first one is the vesicle trafficking, vesicle tethering, vesicle docking, then vesicle priming in some cases, and then finally the vesicle fusion to release the content outside the cell wall. Now looking in detail about the endocytic pathway. Endocytic pathway has primarily three components. One is the recycling circuit. The plasma membrane, the early endosomes or the EEs, the recycling endosomes, and a variety of vesicular carriers. So you can see that these are the plasma membrane which is there. These are the vesicles which are formed, the things are packaged into these, or things are the content comes into these vesicles, and then they can form the early endosome. The fusion of the vesicles will take place to give rise to the early endosome, and from this early endosome, the content which is coming, or the plasma membrane which is coming, is basically recycled and again goes back to the plasma membrane which is there. So this is called as the recycling circuit which is there. That is one primary aspect of the endocytic pathway. Second aspect is the degradation cycle. Now the degradation cycle involves the lysosomes. So they contain the hydrolytic enzymes. So this is the second aspect of endocytic pathway. The third aspect is a connector between the recycling circuit and the degradation cycle, which is the connecting or the feeder pathway. This comprises of what is called as the late endosome. So the connecting or feeder pathway is basically this, the link between the early endosome and the lysosomes or the degradation cycle. An important part of the Golgi network which takes place, which plays an important role in this pathway, is the interacting partner in the Golgi providing lysosomal components is the Tris-Golgi network, which communicates with the plasma membrane. Now this is very interesting that TGN actually interacts with all the three. It can interact with the late endosome so that it gives the endosome all those proteins which are present in the lysosome. So it interacts with the late endosome, it interacts with the early endosome, as well as it interacts with directly with the plasma membrane. So all these three, it can communicate. So this kind of a pathway is when it does not follow the exocytotic pathway. So it can, communicate with the plasma membrane. It can communicate with the early endosomes, as well as it can communicate with the lysosome or the degradation cycle which is there. Now, last one point which I need to make at this point is that the degradation cycle is a shared facility for degradation in the cell, and is not only used for substrates delivered via the endosomes. So this degradation cycle forms the pathway, is shared between many pathways, and endocytic pathway is only one of them where it is shared.
With this note, thank you, ma'am. Thank you so very much for giving us this productive session. Dear friends, we have a lot more for you. You are requested to be with us as we are back after a very short break. Thank you. Hello friends, welcome back to the session. Friends, as you know that today we are talking on our series on cell biology, and we are specifically covering the following areas, that is endocytosis, exocytosis and lysosomes. We have covered a lot and there is a lot more for you to learn about, and for the discussion on the topic we have with us in our studios Dr. Charu Dogra Rawat. Friends, if you wish to ask questions from Dr. Rawat, do call us through our toll-free number. Our number is 1800-110-430. Now I would like to welcome our guest, Dr. Charu Dogra Rawat, once again, and would request her to continue further.
So we were talking about the endocytic pathway, and we talked about the three components of endocytic pathway. One is which is called as the recycling circuit, where the things which are coming from outside needs to recycle back into the outside, which is basically the the plasma membrane which is coming in, or any other thing which is coming in which needs to again further go back outside the cell. Then it follows the recycling pathway which is there. So whatever things are transported from outside into the cell, the transport vesicles fuse together to give rise to what is called as the early endosome, and then from early endosome different vesicles come out that go back and fuse with the plasma membrane to release the content outside, or just the recycling of the plasma membrane takes place. So that was the one component of endocytic pathway. The second component of endocytic pathway is the degradation cycle, or the lysosome. When the things which are coming from outside at the end needs to be degraded, or anything which is coming from the thing, for example the pathogens which are engulfed by a cell needs to be degraded, so they are directed to the lysosome. So that degradation cycle is primarily comprising of the lysosome which is there. And the connector between this regulatory pathway and the lysosome pathway and the lysosome is basically the connecting thing, which is the third component of the endocytic pathway. This component comprises of what are called as the late endosome. So when early endosomes, they pass on the material to the late endosome, and late endosome basically collects the lysosomal proteins, etc., and finally fuses with the lysosome to for the things to be degraded. So they form this is the complete endocytic pathway, and this endocytic pathway we also talked about that the Tris-Golgi network plays an important role, and it actually communicates with the plasma membrane as well as the early endosomes as well as the late endosome which is there. Another point which we made was that the degradation cycle, or the lysosomes, they are a shared facility for degradation in the cell and is not only used for the substrate delivered via endosomes. So this is just one of the pathway, because right now we are talking about the vesicular transport. So this is one of the pathway where the lysosome is involved, but if any other material that needs to be degraded is coming from any other cell organelle, it finally ends up here and is degraded. So now we will look into detail about the different components of this endocytic pathway, which is the early endosome, the late endosome as well as the lysosome. So early endosomes, or EEs, which are called as, they provide the starting point for the late endosome maturation. They were defined initially as the compartment that first receives the incoming cargo and fluid. EEs are now recognized as the main sorting station in the endocytic pathway. So initially it was only known that they are the starting point and they take up the material that is coming from outside, but now their function has been expanded to actually carry out the sorting also. So that is the point where the things which need to be recycled are sent along the recycling pathway, and the things which need to go further, they are sent through the late endosome, and the further, if they are degraded, they go into the lysosome which is there. So these are the two different pathways. So from late endosome, they can go into any other, or the early endosome, they can go into any other part of the organelle also, where the things which are coming from the outside are needed. So these are this is the thing about EEs. EEs membrane and volume is mainly derived from the primary endocytic vesicles that fuse with each other. The EEs are heterogeneous in terms of morphology, localization, composition and function. Looking at this ultrastructure of EEs, most of them are relatively small and patrol the peripheral cytoplasm close to the plasma membrane through saltatory movement along the microtubules. So individual EEs have a complex structure with tubular, as well as vacuolar domain. So these are the intraluminal vacuoles that you can see. So this is one EE, this is the invagination that is occurring, this is the inward budding vesicle which is formed, and these are how they are basically the membrane of the EEs are recycled. This is the way. And this is basically a bilayered clathrin coat, is also present outside the endoplasm, outside the early endosomes which is there. So individual EEs have a complex structure with tubular and vacuolar domains. Most of the membrane surface area is in the tubules and much of the volume is in the vacuoles. So surface area in tubules and the volume content is in the vacuoles which is there. Looking at the late endosomes, the mature LEs are typically round or oval and have a diameter of about 250 to 1000 nanometers. They have a low buoyant density and a high negative surface charge. The limiting membrane contains lysosomal membrane proteins such as called as the lysosomal. LAMP is basically lysosomal associated membrane proteins. LAMP1 and the lumen contains a complement of acid hydrolysis. The lumen also has numerous ILVs, often up to greater than 30, with a diameter of about 50 to 100 nanometers. The pH ranges between pH 6 to 4.9. So what happens in the late endosomes is that the lysosomal proteins which are going to form the part of the mature lysosomes, they are coming from the Golgi network. So they are manufactured in endoplasmic reticulum and they are matured in Golgi complex and they finally end up into the late endosomes. So all the lysosomal hydrolases, etc., they come from the TGN to the late endosomes, and therefore late endosomes contains many more ILVs, which is the intraluminal vacuoles, and they contain these lysosomal associated membrane proteins as well as the lysosomal hydrolases which is there. LEs formed in the peripheral cytoplasm move to the perinuclear area of the cell where they fuse with each other to form larger bodies and to undergo transient, the kiss and run fusions, and eventually full fusions with the lysosomes and pre-existing hybrid organelles between the endosomes and the lysosomes are termed as the endolysosomes. So in the late endosomes, when the maturation is taking place, when the materials are coming which are further going into the lysosome, it does not degrade the material in the late endosome. The materials are only degraded inside the lysosomes or the mature lysosomes which are there. Therefore, these transient vesicles are termed as the endolysosomes, which is the transition between the endosome and the lysosome structure which is there. So by fusing with lysosomes, the LEs follow essentially a unidirectional dead-end pathway, whereas most of the components of LEs are degraded in the lysosomal environment, others are longer lived and contribute to the maintenance and generation of lysosomes. So when the late endosome is going to fuse with the lysosome, then that is the dead end. It just finishes up, entire content is degraded except for the content which has to form the part of the lysosome, such as the lysosomal hydrolases or the lysosomal associated membrane proteins which are there. They remain a part of the lysosome, otherwise rest all the things which late endosomes are carrying, they are degraded inside the lysosome. So you can see the structural differences. The LE contain numerous ILVs and this is an endolysosome with partial electron dense areas, and if you see the lysosome, they have large amount of electron dense area which is there, and lysosomes which is there. So these are images all from HeLa cells that had been prepared, processed for thin sections, EM, and the reference from where this has been taken. So by fusing with lysosomes, the sanctity of the late endosomes is finished, and the proteins which are made for lysosomes are then used in there. So this again comprises the endosome lysosome system, which is now very important. So you can see then from the plasma membrane, the vesicles are coming and fusing together to give rise to this early endosomes. Now this early endosomes can pass on its content to the late endosome maturation, or they can recycle the membrane through this way. So this is the recycling component, or otherwise they will mature to form the late endosomes, and as you can see, they go via this pathway, and at this pathway the vesicles from the trans-Golgi network are coming in and bringing in the different kind of proteins. So you can see that this is a late endosome which is formed. Then this late endosome, when fused with the lysosome initially, then this transient structure is termed as the endolysosome. Now this endolysosome will give rise to the functional. When all the endocytic part, endosome part is gone, then this is basically what is called as the functional lysosome, which contains all mature hydrolases and all lysosome endoplasmic particle which is there. So the lysosome and the endosome, late endosome, fuses together to give rise to this endosome, and then endosome is finished and therefore the lysosome is there. So this is the endosome lysosome system, and it is a very, very important system because we will also look at some of the disease or a couple of diseases which are there if this lysosome system is not working. Then what are the diseases that are created? Before doing that, we'll again talk about the lysosome biogenesis, that now how this lysosome is generated. We are talking in here that the late endosome which is following this endocytic pathway. So we talked about the early endosome, we talked about the late endosome, now they are fusing together. So where does this lysosome come from? So this lysosome is actually coming again from this region only, So how this lysosome biogenesis takes place? So maturation of endosomes is reflected by an increasing number of intraluminal vesicles. So as I told you, therefore in lysosomes there is a higher amount of electron density area that can be found. So you can see this is the early endosome, this is the late endosome, and this is the lysosome, and you can see that there is an increased number of intraluminal vesicles which are there, which is reflected here. So from the late endosome this particular thing is formed, and when there is lot of intraluminal vesicles, and now it is containing the content which is coming from the trans-Golgi network which is there. So lysosomes contain about 40 types of hydrolytic enzymes. For optimal activity, they need to be activated by proteolytic cleavage and an acidic environment, which is established by the V-class H+ pumps on the lysosomal membrane. Why doesn't the lysosomal hydrolysis just degrade its own membrane? When it has the capacity to degrade anything that comes inside it, it must have the capacity to degrade its own membrane. But they are not activated. They are all deactivated, kept in a silent form. They are activated only when they have a proteolytic cleavage or the acidic environment is there. So when this hydrogen ion pumping takes place, or when therefore the fusion of endosome and lysosome is important for the degradation, because when the endosome fuses with the lysosome, the pH drops and this hydrogen pump becomes activated. This ion exchange takes place, and therefore these proteins, there is a signaling mechanism which goes on. So there is a pH which is dropping from the 7.2 to internal pH which is really acidic, and then there is a proteolytic cleavages of the proteins take place which make them activated, and therefore it will degrade all the content of the endosome which is coming inside. Mature endosomes containing numerous vesicles in their interior are usually called multivesicular endosomes or late endosomes. Fusion of a multivesicular endosome directly with a lysosome releases the internal vesicles into the lumen where they can be degraded. Lysosomal membrane proteins are not incorporated into internal endosomal vesicles, thus keeping them away from the degradation. So the degradation is also happening inside these interluminal vesicles. The degradation is not happening in the free lysosomal, lysosomal plasm. It is not happening inside the lysosomal in open. Therefore, the proteolytic or these proteins does not have a chance to, degrade the lysosomal outer membrane. These intraluminal vesicles, they do not break down. These vesicles do not break down because, as I said, because of the pH thing, because of they need to be activated, the proteolytic cleavage needs to occur. So that occurs only when the endosomal fusion takes place. But these in intraluminal vesicles, the lysosomal proteins and lysosomal receptors does not go inside. So these lysosomal proteins, they remain there for a longer time and therefore become the part of the lysosome or all the hydrolases, etc. So this is how this is just a recapture of, what are the macromolecules that are degraded, what are the macromolecules that follow this path. from the early endosome to the late endosome to the lysosome. So they are basically by the phagocytosis or autophagy. So they can be the bacteria which is being internalized. So you can see the bacteria which is coming. They can be any other molecules which are coming inside. So they come inside early endosome, late endosome. You can see here, they can do with the primary lysosome. You can see these are the different mechanisms which are autophagy, a cell organelle which has become useless now. We need to degrade that organelle. So how will that organelle be degraded? So that organelle be degraded by the mechanism which is called as autophagy. So this picture again clarifies the point that this lysosome is not only the pathway of this endocytosis, it is also the pathway of other mechanisms such as phagocytosis, autophagy. Although phagocytosis comes under a part of endocytosis because the thing is coming from outside, but it also forms the part of autophagy, which is the internal cell eating. The organelles which are waste, they are also degraded by these particular lysosomes. So these materials are targeted to the lysosomes for degradation. Now, building a functional lysosome, we are talking about the lysosome biogenesis, and as I said, there are two important components of lysosomal proteins. One is the soluble lysosomal hydrolysis, whose primary role is the degradation, and another is the integral lysosome associated membrane proteins, or what are called as LAMPs, whose primary function is the structural integrity which is there. So you can see this is a lysosome, a typical lysosome which is there. You can see there is a V-type H plus HH hydrogen ATPase, which is a hydrogen pump which is there, and you can see that there are a lot of soluble enzymes which are more than 50 in numbers, cathepsins, which are present inside the cell, inside the lysosome. These molecules, they are lysosomal proteins. Apart from them, there are proteins you can see which are associated with the plasma, which are associated with the membrane of the lysosome. So for maintaining the integrity, the structural integrity of the lysosome, these LAMP proteins are very important. So these are the two. So one has to, when the lysosomal biogenesis is taking place, this kind of an architecture needs to come into place. So there has to be soluble enzymes which need to come. There has to be lysosomal associated membrane proteins which need to be integrated for the integrity, and of course other proteins such as the hydrogen pump or H plus ATPase which is there. So approximately 25 lysosomal membrane proteins are needed and more than 50 lysosomal hydrolases are needed to give the structure of a lysosome. So how does these proteins are trafficked to the lysosome? So we'll talk about the lysosomal trafficking under two heads. One which is the trafficking of soluble lysosomal hydrolases, and another the trafficking of the lysosomal associated membrane proteins. So looking at the trafficking of first these soluble lysosomal hydrolases. Hydrolases are modified by mannose 6-phosphate. So whatever protein that is formed in the endoplasmic reticulum and need to be targeted to a certain organelle has to be tagged. It need to carry certain, a certain modification which tells the Golgi complex when it packages it that, which, or the vesicle that where it needs to go. So the endoplasmic reticulum, these lysosomal hydrolases are formed, but they are modified in the Golgi complex by the addition of what is called as these mannose 6-phosphate M6P region in the cis Golgi. The M6P receptor captures the hydrolases in the TGN as the receptor cycles between the TGN and late endosomes in clathrin-coated vesicles. So you can see what is happening here is, then in rough endoplasmic reticulum, the lysosomal enzymes are formed. In the cis Golgi, a modification is occurring where basically these lysosomal proteins are phosphorylated, mannose 6-phosphate is added to it. These phosphorylated proteins are then packaged in trans-Golgi network into recognized by these particular receptors. So, the sorting has taken place. These are just the other normal proteins which need to be secreted out, and this is a protein which needs to be targeted to the lysosome. So, there will be certain receptors in this clathrin-coated pit which is present, which are mannose 6-phosphate receptors. So, this M6P receptors will recognize this particular protein and will package it into certain different vesicles, the clethorin coated vesicles which are there. In this clethorin coated vesicles, then they will become the uncoated synaptic vesicles, and then they will form the what is called as the late endosome. From the late endosome, the recycling of the receptor will take place. So the recycling of the receptor is taking place between the trans-Golgi network and the late endosome, and we have just seen that TGN actually communicates between the late endosome as well as the plasma membrane, etc. So you can see that the receptor has been recycled here. This late endosome is then going to form what is called as, these basically proteins can come from outside also. These are the secreted lysosomal enzyme which is there. This secreted lysosomal enzyme are following this pathway too, and then from here they are transported into the lysosome which is there. So you can see now this protein which was formed in the endoplasmic reticulum has reached the lysosome by this targeting, which is the modification occurring, which is the M6P phosphorylation, which is targeting this particular protein to be formed a part of the lysosome which is here. So that is how the soluble lysosomal hydrolases are trafficked. So you can see here these lysosomal enzymes modified with M6P are bound by the luminal domain of M6P receptor. M6P receptor lysosomal enzyme complexes are recruited into the clethorin AP1 coated bits. Vesicles deliver the M6PR lysosomal enzyme complexes to the late endosome. M6PR recycles to the Golgi, and the lysosomal enzymes are then delivered to the lysosomes which is there. So this MAP receptor which is coming, it can be recycled outside. It can go out from the outside, recycling of the M6P receptor, and then again from the endocytosis it can be brought in. So it can go to outside. So this is the trans-Golgi network. These are the other constitutive secretion that is occurring. But this is the receptor that has come here. So the receptor which is formed can go into the plasma membrane and can also come into the trans-Golgi network which is there. So this is the recycling of the receptor, and finally this receptor will bind the M6P modified protein and will target it into the lysosome which is there. So it is coming from this particular part as well as it is coming from here part which is there. So the soluble enzymes come into the lysosome from both, one, they are formed from the endoplasmic reticulum, and some of the proteins also come from outside the cell. So the soluble proteins which are coming from outside the cell are endocytosed, and then based upon this M6P receptor they are particularly or specifically targeted to the lysosomes, and then the lysosomal proteins are also formed in the endoplasmic reticulum, from where they will follow the typical the late endosome pathway from the tris-Golgi network to the late endosome, and then from there to the lysosomes which is there. So the soluble enzymes can be targeted in such a manner. Next we come to the trafficking of the lysosomal membrane proteins. Lysosomal membrane proteins require cytosolic signals and adapter proteins for their transport into the target organelle. So they require these signals which are there. The sorting information is contained in their cytoplasmic tail. The majority of LMPs contain in their cytosolic domains tyrosine or dileucine based sorting motifs. These motifs interact with heterotetrameric adapter protein complexes AP1, AP2, AP3 or AP4 that are capable of recruiting the cladrine and thereby initiating the assembly and formation of the coated vesicles. So lysosomal membrane proteins that are formed in the endoplasmic reticulum need to have this particular tyrosine or dileucine cytoplasmic tails, and if they have that, then they are specifically targeted to the lysosomes, and they form the and as I told you, they do not go into the intravesicular lumen. They do not go into the intraluminal vesicles and therefore remain in the lysosome for a longer time and become incorporated into the membrane to form the lysosomal membrane proteins which is there. So this is the overall organization of the membrane trafficking which is there. So this is actually shown between a donor and an acceptor. acceptor, but it can be, from the, this acceptor can actually be the outside the cell. So it can be a cell secretory pathway also. So this is this kind of a directed transport that is occurring. So any protein, the soluble cargo protein that is coming up has to form a transmembrane cargo protein. So you can see that there are certain snares and V snare, etc., which is there. So they form this recognizes the soluble protein, the budding occurs, the coat is formed, and then they are basically excised, the coat proteins are recycled, and this uncoating will then cause the tethering, the docking, in some cases the priming, and finally the fusion. So this is the overall, the vesicular transport system in which we have talked about in detail, talking about the early endosomes, the late endosomes, as well as the lysosomes which are there. Now the last part in here is that when we talk about lysosomes, it's kind of a suicidal bag. It does contain all those hydrolases which can basically degrade any of the organelle which is present inside the cell. So the lysosome is so important that it is basically removing the unwanted materials from the cell also. It's causing the autophagy. So if a mitochondria is not working, it needs to go to the lysosome to be degraded, or any other thing, any pathogen that is coming inside the cell need to be degraded. So lysosome is very important as far as the taking up of wrong materials or pathogenic materials or unwanted materials are coming inside the cell, or if even a cell itself is generating the toxic things, which is autotoxic. Those things are also need to be degraded. So lysosome is a very important organelle. So we quickly go through what happens if this lysosome is not working. What happens, what are the disorders which we talk about when the lysosome is not working. Then lysosomal related disorders, there are more than 50 distinct diseases are caused by a defect in the lysosomal functioning. Worldwide, 1:5,000 births have a lysosomal disorder. So you can, see the impact this endosome-lysosome pathway has. The common symptoms include neuropathologies, kidney malfunctioning, bone deformations, muscle diseases, and a general failure to thrive. For most lysosome-related disorders, there is no cure that is present. Human disorders due to the deficiency of lysosomal function. These are the classes of genetic lysosomal disorders. One is a lysosomal storage or metabolic disorders. They are they form the major component. More than 45 such disorders are there in which the lysosomal storage or lysosomal metabolism, there is a problem. Then there is a pigmentation or there is bleeding. These hydrolases which are contained, as I told you, they are very strong hydrolases, has the capability to degrade anything. So if they start bleeding out from lysosome, there are certain disorders related to that, and there is a non-metabolic without a pigmentation defect. So these are the next classification where they are not related to the metabolic aspect or the bleeding aspect. And then of course there are secondary lysosomal defects, which is such as in the prevalence of cancer, which is there. These are certain lysosomal storage disorders that have been listed here. So you can talk about, and these are the different kind of, so in this case particularly the lysosome has a problem in storing various enzymes and various other things, and therefore all these kind of diseases occur. One disease which I would like to point out is the mucolipidosis, which is also called as the Icell disease. So in this Icell disease, basically this phosphotransferase is not working. So if you remember, we talked about the modification in the cis-Golgi network. So in the cis-Golgi network, the lysosomal enzymes that need to be targeted to the lysosome needs to be modified with this mannose 6-phosphate. So this particular thing is not working, and if this is not working, then the enzymes are not targeted properly. There is no clethorin coated vesicles that are formed, and because of that there is a disease or disorder that occurs which has the characteristics such as growth deficiency, mental retardation, swollen salivary glands, skeletal distortions, etc. The most severely affected system is the skeleton, because when the bone is there, therefore there the leakage occurs, the lysosomal leakage occurs, and therefore it degrades the bone which is there. So ML2 mouse model has also been generated in 2007 to understand this, and you can see that the children afflicted with mucolipidosis, 2 and 3 generally are short in stature and have distinctive facial features. So I touched upon just one of the diseases which is related to the lysosomal disorder. Otherwise, lysosomal storage, lysosomal secretion, it is such an important component that any malfunctioning of the lysosome leads to various kinds of disorders, and there is no cure for them. So I hope this entire endomembrane system involving endoplasmic reticulum, Golgi complex and lysosomes are clearer to you.
With this note, thank you, ma'am. Thank you so very much. Dear friends, keep watching us, keep writing us at info.cec@nic.in. We will be meeting again soon. Till then, take care. Goodbye. Thank you.